Hello everyone, I'm Kaitlyn Casey. I'm a translational biomedical sciences major and a soon-to-be junior at Rowan University. Today I'm here to talk about the research I did as a New Jersey Space Grant Consortium intern. So, for the past decade or so, the pharmaceutical industry has seen increasing potential usage of protein therapeutics for drug delivery. Protein-based biologics are a new, highly useful class of drugs that have been developed from living organisms. They are used to treat a multitude of diseases including cancer, immunological disorders, and heart disease. The current routes of drug delivery for this treatment include inhalation, injection, and both oral and transdermal delivery. This type of treatment has a higher specificity for targets, which means a smaller amount of the drug is required to produce the desired effects, which is great. However, the toll of encapsulating and delivering these drugs in the human body limits the drug's capabilities. In addition to this shortcoming, proteins also have short half-lives in organisms, and due to their high molecular weight, are prone to enzymatic degradation, which decreases their effectiveness in treatment. Recent advances in protein engineering and material science have made the targeted delivery of enzyme therapeutics using nanocarriers a promising new model of treatment. Emulsification techniques are used to encapsulate molecules in nano-sized particles. The optimal vehicle for this therapy will be a nano-sized particle that allows the drug to target specific tissues, preserve the original 3D folded shape of the protein, and prevent degradation from the human body. In addition to finding an optimal method for drug delivery, encapsulating the drugs also provides a great way to package and store the drugs for long periods of time under stressful conditions. This could be extremely useful in the future for space travel. There are some hybrid approaches to creating solid nanoparticles, which allow for a large aqueous core and enhanced malleability. Water in oil and water nanoemulsions offer the ability to successfully encapsulate therapeutically relevant concentrations of biologics. Biomacromolecules respond better to the production process of water in oil and water because high pressure is not necessary for emulsification, which is why this is the best approach. One way that drugs can be encapsulated is through the use of reverse micelles. Reverse micelles are a water and oil nanoemulsion that use surfactant molecules to disperse aqueous solutes in non-aqueous solvents. Our mixture is proven to encapsulate a wide range of macromolecules while fully sustaining their structure. Because of this property, we are exploring the potential of this mixture as a drug delivery formulation, particularly for challenging drug delivery problems, including the delivery of protein-based therapeutics. Formulations were created using various ratios of surfactants and water in different oils. The formulations included reverse micelles made from surfactants and encapsulated specific dyes. We tested these reverse micelle formulations on both the immortal mammalian cell line HeLa cells and Saccharomyces cerevisiae, which is just regular yeast. To test whether the reverse micelle formulations made are lethal to mammalian cells, we used the cell titer blue cell viability assay. This test was developed as a way to determine the number of viable cells present in a given sample. First, the HeLa cells were exposed to the formulations, and then the cell titer blue dye was added, which contains the dye residurin, which is metabolically reduced to resirufin in viable cells. Resirufin is highly fluorescent at 573 nanometers, allowing for the detection of metabolically active cells after a sufficient incubation period. When HeLa cells are exposed to the cell titer blue reagent, viability can be observed as pink cells, while metabolically inactive cells remain a dark blue color. Precise data is collected using a plate reader that reads for the fluorescence given off by each well. We tested both the samples containing the solvents and reverse micelles and samples with just the solvents and propynium iodide to see if it was the reverse micelles themselves that were lethal to HeLa cells. In a few cases, it did appear that the reverse micelles had a negative effect on these mammalian cells, but there were samples in particular containing lauryl glycol 90, labrafact PG, and pluralolic that were not lethal to HeLa cells in the presence of reverse micelles. When tested for lethality on the yeast, these solvents were also found to be non-lethal, whether they contain reverse micelles or not. The yeast was tested for lethality by being exposed to these formulations and then plated on a YPG plate and observed for growth. We also performed this assay to determine if these formulations were able to permeabilize the yeast. After the yeast was exposed to formulations containing propynium iodide, which is fluorescent, it was read by the flow cytometer, which could see if the PI in the formulations had managed to breach the yeast membrane. These findings led us to believe that most of the formulations that were non-lethal were not killing the HeLa in yeast because it was not managing to actually permeabilize the cells. The formulations, such as labrafact PG and pluralolic, were most likely sticking to the outside of the cells or floating in an aqueous layer. However, oral glycol 90 proved to be an extremely effective candidate for drug delivery because it managed to both permeabilize the cells and keep them alive. Reverse micelle encapsulation is also extremely effective for packaging and stabilizing drugs and solvents. It's a great candidate for keeping drugs safe in stressful conditions, which would make this kind of encapsulation extremely helpful for both clinical and laboratory settings, as well as space travel. I would like to say a huge thank you to my mentor, Dr. Carone, as well as Dr. Nucci and all of the people in both of these labs that helped me with this project. I would also like to thank the people from New Jersey Space Grant Consortium who gave me this great opportunity to perform my research. Thank you so much.